Great. Welcome, everyone. It's a pleasure to be here. I want to thank the organizers as well for the opportunity to provide an update on Miromatrix. So for those of you who are new to the Miromatrix story, our mission is to eliminate the organ transplant waiting list. So kind of going on the similar theme from the last talk. But our focus is really how do we solve one of these largest unmet needs? If we look out today, we know that over 120,000 patients today are waiting for an organ. About 70,000 of those patients will not receive the life-saving technology or medical need that they need this year. So we've developed a way to essentially bioengineer transplantable organs with a focus on liver and kidney. Currently at the IND stage, we filed our IND last year, currently on clinical hold, but looking to do a full response to that hold letter by the end of this year. And I'll get into that a little bit more. We've got great strategic investors as well as scientific collaborations. Here's a quick video, though, that just kind of gives a quick overview of our technology and our approach to bioengineering transplantable organs. Our patented perfusion decellularization technology starts with a functional organ from a pig that would normally be discarded. Next, we remove all the organ cells by first cannulating the organ and then flushing it with a simple solution that only removes the cells, resulting in a pristine decellularized organ matrix that can be stored for over 12 months. Next, the decellularized organ is placed into a bioreactor, and we initiate our patented recellularization process. During this step, human cells are introduced to the organ, and over the next 14 to 28 days, the organs are continuously monitored and perfused with the appropriate nutrients and oxygen to achieve the desired key growth parameters, resulting in a bioengineered organ. It's a really nice overview. If we break down our process, it really consists of two aspects of it. First is that decellularization process. That's the ability to take an existing organ, cannulate it, perfuse it with a mild detergent, and rapidly remove all the cellular material. Now, to start with, we wanted really an unlimited source there. So we start with a porcine material from that. Great historic references within the medical device community to use porcine tissue, but it was unknown if we could use our process in it. So we actually took whole porcine livers, perfusion decellularized those, and created a surgical mesh called Miromesh and a wound care product called Miroderm, which we got 510(k) clearance and have since transplanted that and treated thousands of patients with that. So we've got a really good safety profile associated with the base material. The second portion of that is really focusing on how do we recellularize it. So that's the introduction of human cells, as you saw inside the video, to recapitulate and bring that organ function back. That, we believe, allows us to bioengineer organs with a key differentiator in here that I'll talk about in a second, and that is we believe and have really received confirmation through our IND process that the FDA doesn't view this as a xenotransplant, because we no longer have any animal cells or animal serum or anything left inside of the matrix, and it's really all human cell based, highly scalable, and utilizes the same transplant techniques that are utilized today for whole organ transplant. If we do look across this, as I mentioned in the beginning, there's a tremendous need, right, for these types of organs. So we're not alone in trying to develop therapies. Today, human to human, we know that that's a gold standard. The challenge with this is there's not enough organs. Last year alone, there was about 25,000 kidneys that were transplanted overall, about 40,000 of all the organs. But we know that we have about 120,000 patients, again, waiting for an organ. So while that works well, and we know we can immunosuppress those patients, there's just a tremendous lack in the need, or it's tremendous lack in the supply. So an alternative technology that you probably heard a lot about is xenotransplantation. That's the ability to potentially genetically modify the pig to go directly into humans. Certainly that's been in the news and continues to make progress. I think two of the challenges in there where we differentiate ourselves is, one is the ability or the concern associated with transmission of porcine viruses. And we saw that in the initial heart transplant patient from last year. And the other one is a large unknown on the immunosuppression required to move forward. Our solution to that, and I'll get into that a little bit more in a second with bioengineering, is by taking the scaffold and then repopulating it with human cells. We've already demonstrated that we have viral clearance in our first commercial product that we had to commercialize. That really provided the safety profile. And the second portion of that, since we're now taking a scaffold, adding cells onto it, with human cells, we believe that the immunosuppression for these products should be similar to the gold standard today for a deceased donor or living donor organ transplant. So if we get into our process a little bit more before into the data associated with it, the recellularization is a fast process. Once we decellularize the organ, it's incredibly stable. We place that into a bioreactor like you saw on the video. We then introduce human cells into that, and that process takes about 2 to 4 weeks to recapitulate that matrix and get to a level that we believe it has the appropriate functionality. The lower middle picture there is actually a revascularized kidney that's being transplanted back into a pig at Mount Sinai. Question we get a lot is what type of cells are you recellularizing these with? So in our first generation product, we actually take whole organs that weren't placed for transplants. These are organs that could have been placed for transplant, but for whatever reason, bad biopsy, long ischemia time, long transport time, they weren't placed. That allows us to source those organs into Miromatrix, and now we isolate out the primary cells. So we'll isolate out primary liver cells or isolate out primary kidney cells and place those back onto the matrix. As part of that, we see an expansion factor. So today, 1 liver gives us up to 4 liver grafts. And more importantly, on the kidney side, 1 kidney, we easily see 8 or more kidneys that we're able to recellularize using those isolations. So there's a nice expansion factor. Our whole premise here is to start with something that's the simplest, and that's why we're starting with primary cells derived from these organs, because that really allows us, we believe, the fastest path to being able to treat patients and providing this life-saving technology to them. Once we're there, once we're able to demonstrate that, then you can foresee a future generation where now we're doing this with stem cell therapy to really eliminate the ultimate need of immunosuppressive therapy. But we're really focused on how do we step through this step by step. Early last year, we opened up our manufacturing facility. So not only do we have the ability to do this, we do everything in-house. So we have a full manufacturing facility that allows us to complete our development, but also do phase 1, phase 2, and up to phase 3 clinical trial material in-house. As I talked about before, as we look at how we go from this and get to our ultimate goal, we've really looked at how we de-risk this and continue to move through the technology. It started with the decellularization of the whole organ, creating a medical device from that and demonstrating the safety profile for that. We've since out-licensed that to Reprise Biomedical. The next step, and what I'll talk about next, is the product that we're currently at the IND stage as, and that's before we go fully transplantable with the liver and kidney, is the ability to take the bioengineered liver, house that external to the patient, and essentially create liver dialysis. For the acute liver failure patients, these are patients that today have no alternative therapies outside of transplant. 30% of these patients, unfortunately, die. 25% get a liver transplant, and 45% can recover. And then once we're able to achieve that milestone and gain that clinical data, we're then able to utilize that and that ability and what we learn from that process towards our fully transplantable liver and kidney programs. And then here's just a quick video that just gives a sense of what is Miroliver ELAP. How are we treating these patients with an external liver assist?
The Miroliver ELAP system from Miromatrix is a revolutionary external liver assist product. The system consists of our bioengineered liver and a commercially available external blood pump. The patient is connected via tubing to the system. The system then facilitates the movement of blood from the patient throughout the bioengineered liver and back to the patient. The bioengineered liver is designed to provide temporary liver function for the patient, essentially providing liver dialysis. Treatment can be provided for a number of days.
So hopefully that gives you a good sense of our lead product that we continue to move forward on. As I mentioned with that, the Miroliver ELAP, we're currently at the IND stage. We filed our IND last year. We're placed on clinical hold and are getting ready to do that full response by the end of the year. We're hopeful that we then get clearance and that we're in human clinical studies early in 2024. Our next 2 lead programs, Miro Kidney and Miro Liver, are behind that within the pre-IND stage before we go forward and submit the IND on those programs. So real quick, what is the data in terms of what we're achieving and what have we demonstrated to date on these programs. So if we take a step back for a second and we say part of the recellularization process, the key first step is to demonstrate not only that we can take the matrix, but can we put vascular cells back in there, essentially revascularize the construct and solve the vascular challenge, because without that, you can never get the more advanced cells back into there and be able to create that final product. So here's something that we published in Nature Biomedical Engineering a few years ago. This is our ability to take that revascularized liver, transplant that back into a pig model, and you can see once we take the clamps off the portal vein, you're going to be able to see that initial inflow into the graft. It's still going to be white up on the top because we haven't taken off the outflow clamps. Once we take the clamps off the cava there, now we have complete perfusion of this graft. So now we've actually created a construct now that we can anastomose in and integrate into the circulatory system of the host. This really allowed us to move beyond simple tissues to something that is more complex and allow us to move forward with bioengineering the organ. The next aspect of that is can we demonstrate liver function? So now we take whole organ, whole liver isolates, recellularize that, and demonstrate that we can bring that liver function back, both from a standpoint of maintaining the endothelial function with VWF, but more importantly, we can show albumin production, we can show urea production, and ultimately we can show ammonia clearance, which is the one unique function, one of many unique functions of the liver, where we can dose in high doses of ammonia, and we see beautiful clearance associated with that. Not only that, we've demonstrated all sorts of functionality and sustained functionality of this, whether we're looking at ammonia clearance, urea production, fibrinogen production, A1T production, and we do this over a number of days, as well as demonstrating not only that we can achieve that, but we also can put these into cold storage, simulate shipping to the clinical sites, and demonstrate continued functionality of these grafts over the duration of what we'd be looking at from the therapy. Once we've demonstrated that we had that functionality at that level, we also created a large animal acute liver failure model where we essentially now transplanted in, instead of keeping it external, looked at it internal, and we've published on this recently as well, to demonstrate that now we could ligate off the native liver. Volumetric CT scan shows the absence of any flow to the native liver, and you can see our bioengineered graft just above the kidney inside of this. And what we see with this is inside the control, similar to what you'd see with acute liver failure, high levels of ammonia. Eventually, this is where patients go into a coma and eventually die. But in our bioengineered grafts, you can see that that ammonia level rose, but then leveled off, in some cases is coming back down. Since then, we've increased the size of our graft and the overall load of hepatocytes in there. So not only could we demonstrate on the bench that we have liver function, now we're seeing that liver function in vivo as well. This really set us up to move forward with our first planned clinical application of our technology, which we've described as the Miroliver ELAP. As I mentioned before, 30% of patients die from acute liver failure, 25% receive liver transplants. We're really looking at this ability of this, because the liver is the one organ that can regenerate itself, is that notion, at a minimum, can we bridge the patient? Can we provide a longer duration to allow them to receive a liver? Our true hope of this, because the liver is the one organ that can regenerate it, can we bridge it long enough, actually a bridge to recovery, to allow these patients in to recover and no longer need a liver transplant, and free those livers up for end-stage liver failure as well. So excited for this. As I mentioned, we're in the process of responding to the FDA on our clinical hold on this. And what's really exciting about this is a fast study. We're looking at 15 patients. This is up to 2 days of continuous therapy. So primary endpoint at 2 days, secondary endpoint at 21 days. So we look to enroll this rapidly. It's a plan for a multi-site clinical study. In the minute that I have remaining, some of the excitement stuff that once we move this forward as well is just to give you a preview a little bit on the kidney side. So we can demonstrate the same things with kidneys. We can isolate out, we decellularize them, we recellularize and populate them. We can demonstrate that we can repopulate the vasculature, more importantly the glomerulus, which is the filtration substrate of the kidney, and then the nephron. Similar to what we showed with the liver, we also can recellularize that, transplant that back in, and demonstrate we get long-term perfusion in a large animal model. So again, solving that vascular challenge. And the next thing that we demonstrated and announced early last year as well, was the ability not only to revascularize, but now we can show the functional recapitulation of the glomerulus, and more importantly, active filtration of that. And you can see some of the urine production there, and the ability to selectively retain high molecular weight proteins and other things. So that program continues to move along nicely as well while we focus on our lead program. So that's a quick update on Miromatrix, where we're at, the excitement that we're looking at. As I mentioned before, our whole mission is to bioengineer transplantable organs to eliminate the transplant waiting list. We see a different future for transplant, and excitement around bringing tissue engineering forward, where the patient's no longer waiting for that call, but how do we actually create organs where we can schedule these in advance and save lives. So appreciate the time. Thank you.